I'm Dr. Tim Inglis, and I'm here today to tell you about the diagnostic work that PathWest Laboratory Medicine does in conjunction with the University of Western Australia and the FAST Lab, which is a collaboration by the two. I'm a medical microbiologist employed by PathWest, and I also head up pathology and laboratory medicine at UWA. Second slide. What I'm going to talk about today is the practicalities of getting diagnostic tests out through the regional laboratory network to support the Country Health Service. I'll give you some general perspectives on some of the diagnostic challenges, and then I want to have a look at a number of key applications, particularly meningitis, septicemia, what we've done overseas, what we've done bush, and no such talk will be complete without some comment about COVID-19. Slide 3. So some key considerations that we need to take into account are that we've got a good regional laboratory network here in Western Australia. It's a whole network of clinical laboratories, usually in larger regional hospitals, but many of these are thousands of kilometres away from the Perth metro area where most of the big laboratories are located. Regional labs have to cover all acute pathology services for their hospital and have to do so with a small staff. And currently, there is no significant molecular diagnostic capability outside of the Perth metro area. Now, a few things about diagnosis. When we talk about a diagnosis, it's actually usually made by the clinician dealing with the patient, either in the clinic or on the hospital ward or maybe the emergency department. So the usual diagnostician is the physician who assesses the patient, not the laboratory. The laboratory has an important support role, and it addresses three key questions: how sick the patient is, why they're sick, and what then we need to do about it. Slide. The rapid methods that we use and that we're familiar with are ones that address each of these questions, and the particularly rapid bedside tests, the point of care tests that we think of have a very limited role in this sort of activity. So what we do most of the time at the moment is we use for infections the peripheral white blood cell count and the C-reactive protein. Concerning why the patient is sick, in clinical microbiology terms, that is the identity of the microbe that's causing the infection. And most of the work that we do here uses a microscope, or it may use antigen tests. Occasionally it uses some of the big equipment that we got in central laboratories in the Perth metro area, but not in the regional labs. And when it comes to working out what to do for the patient, in infection terms, the key information is what antibiotics to use. And our antimicrobial susceptibility tests are what guides the choice of treatment. Next slide. The specific challenges of our remote communities in Western Australia are clearly that the distances are very large for specimen collection, delivery and transport. They're also large for supplying diagnostic equipment and diagnostic tests, maintenance and for troubleshooting of that equipment. So these are some of the reasons that it's very difficult to do work with the same level of complexity and intensity as the Perth metro area in regional centres. But also the demand for any specific tests is sporadic. It may be unpredictable if the numbers are very small, and therefore it's difficult to know what reagents to have in at any one time, especially if they have a very short shelf life. The duration of a remote laboratory service may be very long and therefore open-ended. And as you may realize by now, the pathology laboratory staff in the regional centres have to multitask to a level that is not seen in the big clinical labs in Perth. Any reagent wastage can be costly, and this is particularly so in regional centres, where some of the tests that we're talking about may run to more than $100 a single test. And so if a box of those tests runs past its use-by date, that may be very costly and run to several $1,000s of waste. Quality control is also very tricky in remote areas because all of these things have got to be done within a quality control system, and meeting those conditions can be very challenging at a long distance. Slide 6. Let's consider what can be done about meningitis. The first question, how sick is the patient, is not something we've got a specific test for, but clearly a patient with meningitis is going to have some very clear-cut clinical signs, and the doctor looking after the patient usually is very clear about the seriousness of the patient's problem. So we move on very quickly to working out why the patient is sick, identifying the organism responsible. Now, there's been a big development in the diagnosis of meningitis in recent years. We're now using rapid molecular tests based on PCR. And there's one such test that has been validated by us here in the Perth metro area, and specifically at the Perth Children's Hospital. It's called BioFire, and it uses a panel of tests to give us answers to 14 different causes of meningitis in about an hour from receiving the specimen to the lab. This equipment isn't yet available for routine use in regional Australia, but we have tried it out at the Children's Hospital, and we've used it for other purposes in regional centres. So we're confident that this could provide rapid answers to meningitis if the equipment was available in each of the major regional centres. What to treat the infection with is something that is guided to a certain extent by the results of real-time rapid PCR, but actually getting a clear indication of specific antibiotic sensitivities takes additional time. So there is some development work going on at the moment to try to speed that up. Next slide, slide 7. And that brings me on to a second application. Blood culture is used to identify the organisms responsible for bloodstream infection. It takes 24 hours or so to get an initial result and 48 to 72 hours to get antimicrobial susceptibility results. That time is a luxury you don't have if you're working in a regional hospital with a patient who's got a fever and other signs of sepsis. And so the BioFire system has been developed to give a very rapid answer on a blood culture when it first comes up positive in the laboratory's autoanalyzer. And we've demonstrated in Broome and Bunbury that it's possible to reduce the time to specific identification and therefore indication of what antibiotics to use in bacteria that grow in blood cultures when you use the BioFire. This is not yet routinely available, but once again, this PCR-based system would be suitable for major regional centres and anywhere big enough to be running blood culture equipment. greater than 24-hour time saving means patients are less likely to deteriorate. The disease will be treated with the right antibiotics before the patients have got worse, and therefore there's a better prospect of patients getting out of hospital faster, and for severe infections, possibly patients surviving at a higher rate, which is always a good thing. Next slide. Slide 8. We've taken some of our PCR diagnostic tests to distant locations in countries like Sri Lanka and Malaysia. In Sri Lanka, we started in 2008 with a World Health Organization twinning project between PathWest and a university in the center of Sri Lanka. We did a series of training workshops, and with support from Rotary International, we installed quite a lot of molecular diagnostic equipment in centers outside of Colombo, the capital, and those are now being used for diagnosis of exotic infections that are present in the tropics, also present here in tropical Western Australia. Meningitis was one of those infections, but also they have the ability now to achieve a confirmed diagnosis of dengue. Slide. We've applied this regional deployable PCR approach to tracking down arbovirus infections in remote places in northern Australia. We first did this way back in 2010 in far north Queensland during a Defence Force exercise in the High Range National Park area, and we were able to trap mosquitoes and same day work out what arboviruses such as Ross River and Murray Valley those mosquitoes were carrying. And then 2 years later, we did an end-to-end drive at the Gibb River Road, trapping mosquitoes along the way and testing them. And we were able to demonstrate arboviruses even during the long dry spell when there are fewer mosquitoes around. So these things can be done in remote places, and environmental health threats even can be assessed out of the back of a four-wheel drive. Next slide. Clearly, I had to say something about COVID-19, and as we've recently had to do a lot of work on this, you see here a map, and this shows you the campaign to deal with COVID in regional Western Australia. The symbols at the left-hand side show you that under the authority of HQ, which is the Public Health Emergency Operations Centre, various different laboratories, PathWest, Western Diagnostic, CliniPath, have been doing diagnostic work on those COVID swabs and have been supporting COVID clinics. There's also a little microscope in a box on wheels, and that is a mobile COVID diagnostic test capability. And as you're well aware, the Gene Expert system has been already deployed to a number of regional centres, including Broome and Kalgoorlie, and further centres are planned in the near future. These small-scale diagnostic activities are capable of handling one cartridge at a time, up to 4 tests simultaneously. They're not suited to high throughput work for mass screening, but they are at least available for rapid diagnostic purposes. And though it has been attached to the COVID-19 pandemic, this has given us an opportunity to try molecular diagnostics in regional Western Australia and demonstrate that PathWest can bring tests closer to where the patients are. Next slide. So to round this off, in summary, we have got some significant obstacles to offering rapid molecular diagnostic capability in regional Western Australia, but these are mainly logistic issues, and they can be overcome if circumstances demand. The main tasks that clinical laboratories achieve is support to the diagnostician with the scientific evidence that we provide for how sick the patient is, why they're sick, and what has to be done to treat them. The options that we've traditionally relied upon have been microscopy, a conventional gram stain, or alternative stains for some specific infections, antigen detection, one or two other rapid tests. But in the Perth metro area, we're heavily reliant now on molecular diagnostics like qPCR, and we're beginning to use some flow cytometry techniques. While this has been very limited outside the major centres, COVID-19 has given us an opportunity to give this a second thought and to reconsider the potential for molecular diagnostics in regional WA. And really, to conclude, culture-independent tests are the priority if we're going to get rapid diagnosis of serious infections in regional Western Australia. Validation and field trials of multiplexed PCR arrays, such as the BioFire system and the Gene Expert for blood culture and other indicators of severe infection are planned for the future. The suitability for regional locations is something that COVID-19 is giving us an opportunity to assess. And finally, I leave you with some references. Obviously, I haven't been able to meet you face-to-face. I'm very happy to take some questions by email. But here are some references you might like to have a look at that will give you some insight into how we have used molecular diagnostic methods in remote locations in Western Australia and beyond. I hope we're going to have an opportunity to work together in future, and I certainly hope that we're going to see a big expansion of rapid diagnostic capability for infectious diseases in regional WA in the years to come. Thank you for your attention.